In this brief presentation, we're going to look at how solutes and solvents move through cell membranes. Molecules in a fluid, such as air or water, move in random directions, bumping into each other as they go. This random motion is called Brownian motion. Despite this randomness, molecules tend to move from regions where they're highly concentrated to regions where they're less concentrated. For example, if you open a bottle of perfume at one end of the room, the molecules of perfume are highly concentrated around the entrance to the bottle. Random motion of the molecules will tend to disperse them until some of them have reached the far corner of the room. This is similar to racking pool balls in the middle of a pool table and then shaking the table. After a bit of shaking, the balls have not been directed to any one location. However, they've been moved from the middle and are now evenly distributed throughout the table. In addition to moving randomly through an open space, molecules can also diffuse across a membrane. Like with an open space, they'll diffuse from regions of high concentration to regions of low concentration. We refer to this as diffusion down the concentration gradient because they're moving from high to low concentration. As you can see in this picture, the membrane sort of acts like a sieve. That is, it allows some molecules to move through, but not others. When there's a strong difference in concentration, the molecules will tend to move from the concentrated region to the less concentrated region, or down the concentration gradient. As diffusion continues, more and more of the orange dye molecules will appear on the right. When they do so, some of them will start to move back from the right to the left. However, the net diffusion will still be from left to right because that's going down the concentration gradient. Eventually, the concentration on both sides of the membrane will be the same and the solution reaches an equilibrium with no net diffusion. When we have two different solutes, each one of them behaves independently of the other. You can see here that the purple molecules are more concentrated on the right and their net diffusion is toward the left. Eventually, like orange molecules, they'll also reach their equilibrium and move back and forth across the membrane equally in both directions. A special case of diffusion happens when we're talking about the movements of the solvent rather than the solute. In biological systems, this almost universally means the movement of water. If you separate two solutions with a selectively permeable membrane, water may pass from one side to the other, but solutes may not. If the two solutions differ in the amount of solute, then there will be a net movement of water in one direction. Let's step through that to make sure that we understand correctly which way the water will be moving. In this figure, the solution on the right has more sugar molecules dissolved in it than the solution on the left. Sugar is more concentrated on the right and less concentrated on the left. Because the water molecules form a hydration shell and dissolve the sugar molecules, it's as if there are fewer free water molecules remaining on the right than on the left. Therefore, when water molecules move through the membrane, they will move from the region of high concentration to the region of low water concentration. In this case, they're moving from the left to the right. This is often confusing because we usually think of concentration in terms of the solute rather than the solvent. Sugar, the solute, is more concentrated on the right, but water, the solvent, is more concentrated on the left. Since it's the water that's moving, it is the water that moves from the region where it is more highly concentrated to less concentrated. As you can see here from these experiments of the U-shaped tubes, water is moving from the left to the right. In fact, the osmotic force is strong enough that one side of the U-shaped tube will have more water than the other. Eventually, gravity will push back, preventing more water from going into the right, and an equilibrium will have been reached. Let's see how osmosis affects animal cells, but before we do, we need to address a little bit of vocabulary. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. Now, when we think about tonicity, we always have to be comparing two different solutions separated by a membrane. It only makes sense to say that a solution is hypotonic in comparison to another solution. The solute concentration itself is not enough to know whether water will go in one direction or the other. You must know the relative concentration of solutes, not the absolute concentration. In order to keep these terms straight, I like to think of tonicity simply as the concentration of solutes. Thus, a hypertonic solution is a solution that has more solutes than another solution. If you place a cell in a solution that is hypertonic compared to the cell, the solutes are more concentrated outside the cell, but the free water is more concentrated inside the cell. Water will therefore leave the cell by osmosis. If a cell is in a solution that is hypotonic compared to the cell, the solution has few solutes and therefore more free water than the cell. Water will therefore move into the cell. Finally, in a solution that is isotonic to the cell, the solute concentration is equal inside and outside the cell, and there will be no net movement of water either into or out of the cell. If you put an animal cell in a hypertonic solution, water will tend to leave the cell. But if you put it in a hypotonic solution, water will tend to enter the cell. Because animal cells have no rigid cell wall, they will expand and may burst. If you do the same with plant cells, the result's a bit different because plant cells have that rigid cell wall. If you put a cell in a hypertonic solution, water will leave the cell, particularly from the central vacuole. The cell will not collapse because the rigid cell wall will hold it, but the volume within the cell membrane will be much less, and the membrane will pull away from the cell wall. This often is seen as a green circle in the middle of a cell, and this is because all of the chloroplasts are now clumped closely together. This effect is called plasmolysis. If you place the plant cell in a hypotonic solution, such as fresh water, then water will tend to enter the cell. As the cell swells and the plasma membrane pushes against the cell wall, however, because the cell wall is so rigid, it does not burst like an animal cell. This condition of being full of water allows plants to avoid wilting, making them what we call turgid.